of the molecule polar. All right, so moving on to our third macromolecule, proteins. Proteins are built up or built upon amino acids. And here we have a central carbon atom, and we know that carbon can form 4 single bonds based upon what we learned back in chapter 2. And all amino acids have a central carbon with a hydrogen bound. They also all have an amine group, which we learned is one of our functional groups. They have a carboxyl group, which is another one of our functional groups. And then they have a third group that's called an R group or a variable group, or the R standing for the rest of the molecule. So there are 20 different amino acids found in living organisms. All 20 are identical except at the R group. The R group is what makes each amino acid unique, and each R group has its own unique features. Some R groups are polar, some are nonpolar, some are charged, some are not charged, some have sulfhydryl groups, some don't. So when you string amino acids together to make proteins, all aspects of the protein are the same except the R group. So in this example, we can see 2 amino acids, methionine to the left and serine to the right. The methionine has as its variable group 2 CH2 groups, a sulfur group, and then a methyl group CH3, whereas the serine has a CH2 group and then a hydroxyl group. We bind these together through a condensation reaction, and through the process that we'll learn in the next week, we are able to continually bind amino acids together, which you can see all the way over to the right, methionine bound to serine, bound to arginine, bound to glutamine. So we build a polypeptide or a protein from assembling amino acids. And what makes each amino acid unique is their R group. Once these amino acids are assembled into a linear chain, that is known as our primary structure of a protein. What determines this is our DNA, and again, we'll learn this in future chapters. But our DNA codes for amino acids. So depending upon the order of coding for those amino acids, we will have a different primary structure to a protein. So the primary structure is basically the number of amino acids, the order of the amino acids, and the kind of amino acids. And by kind, I mean what's their R group. And that's what gives them the different names, lysine, glycine, and arginine in this example. Secondary structure is built upon the primary structure. And secondary structure is where you have folds or twists or coils forming in the polypeptide chain, and these are held together by hydrogen bonds. That secondary structure then starts folding back on itself, creating what's known as a tertiary or a third level of folding. And this third level of folding is held together not just by hydrogen bonds, but by disulfide bridges, ionic interactions. Maybe a positive R group is next to a negative R group. Hydrophobic interactions. Let's say you have a group of amino acids that have R groups that are nonpolar. They're going to hide from the watery environment of the cell. So the tertiary structure starts to make pockets and three-dimensional shapes that starts to determine the functional nature of the protein. And in fact, some proteins stop there at that level of folding. But others have the fourth level of folding, known as the quaternary level of folding. And as you can see in this illustration, the quaternary level of folding is made up of 2 or more polypeptides bound together through a various means of binding. It could be ionic bonds, it could be hydrogen bonds, it can be covalent bonds. But if a protein needs other polypeptides to be functional, it will have the fourth level of folding. And we can see in this illustration hemoglobin, which is the polypeptide that carries our oxygen in our blood, in our red blood cells, and it needs a total of 4 chains to be functional. Here's another example of proteins, and in this case, they're showing you the keratin in an individual's hair, and that keratin is made up of large aggregates or groupings of proteins. Proteins have a variety of functions, from being a transport molecule like hemoglobin to being a structural molecule like keratin in our hair to being a catalytic molecule like enzymes throughout our body. And the last type of macromolecule we have are nucleic acids. And nucleic acids are built upon nucleotides. Nucleotides are made up of a 5-carbon sugar, which you can see is the orange represented molecule at the top of the slide. They're made up of a nitrogenous base, and there are different types of nitrogenous bases, but at the top molecule it's represented by the blue shapes. And then they're made up of phosphate groups, which you can see as the golden yellow shapes. So each nucleic acid, DNA or RNA, are built upon nucleotides. Now you can also have just a nucleotide exist by itself and be functional, and that's what the representation is at the top. That is adenosine triphosphate, or ATP, which is a functional nucleotide all by itself. That is our energy carrier of our cells. It also can become a part of a more complex nucleic acid called RNA. In the illustration in B, we see a chain of nucleotides held together by covalent bonds. And then in the illustration C, we see a very complex molecule. That's deoxyribose nucleic acid, or our DNA. So again, a recap. Here we have a nucleotide. We see the 5-carbon ribose sugar, the one with the little apostrophe, that's called a prime. That's a numbering system for the carbons. So we can see there are 5 of those in the sugar ribose. We can see attached to the first carbon there is a nitrogenous base that's made up of 2 nitrogen rings. You can have nitrogenous bases made up of only 1 nitrogen ring. And then attached to the 5th carbon, we see 3 phosphate groups. Again, here we see that those nucleotides can be linked linearly and covalently bonded, forming a nucleic acid. I hope that this presentation has been helpful. That concludes Chapter 3. Please stay tuned and look for Chapter 4.